All right, this is part 4 of our notes and the very last part before we turn them in. So once you finish this portion, everyone should be on page 10, slide 31. So we're going to learn about microscopes, and this is just introduction here. And then after this, we're going to have an online microscope activity, okay? So how do we know cells exist? Well, go back in time, a couple hundred years ago, and the first microscope was invented, and they saw what looked like jail cells, one after the other, all the same shape, in a plant, in cork from a tree bark. And so they called them cells, and that's where that name came from. But that wasn't possible to do without microscopes, without being able to see beyond what the human eye can see, right? Okay, so the kind of microscope that we're going to be utilizing in our virtual lab is the compound light microscope, just like this one, okay? You're going to have to label this and everything. I believe on the back page there's a labeling. Yeah, so pay attention so that you know all the parts, all right? So 2 lenses to magnify. One is here, the ocular lens or objective lens, and one is here in this portion of the microscope right there, okay? And yeah, all right. So specimens must be viewed through very small space. They can't be large specimens. Like you can't put a shell like this. You can't look at something like that through this type of microscope. This would have to be something smaller or put thin on a slide. Okay, like somewhere in here. I thought I had some slides, but maybe like this one. Okay. Like this. So this is the kind of thing that you would look at. Very thin, very small, and you may be able to see it, but not see its parts with the naked eye. So in order to get close, you put that, you've all used these before, you put it on there, and you adjust all the parts. You're going to do all this on your virtual lab. And they can be viewed alive. That's a good thing about this. So you can put living things, living cells, living tissue on that slide and view it up close. And you can use stains, but they often kill the specimen, unfortunately. So if you were to take some epithelial cells from the inside of your cheek with a tongue depressor and a piece of wood, popsicle stick, right, and put that on a slide, you wouldn't be able to see them very well. But and they'd be alive still. But if you put dye on them, a stain on them, to make them stand out more so you could see the parts, it would kill the cell, just, through the process. All right, so here are some visuals of cells that you'd be able to see. So types of things you'd be able to see with this type of microscope. So this is a replicating cell. Here are some algae, and so this is phytoplankton and zooplankton. So zooplankton are animal-like, phytoplankton are plant-like microscopic organisms. Here are some more paramecium-like organisms, and here are bacteria replicating, or actually they're being eaten. The bacteria are being eaten by a white blood cell. Look at that. So these are white blood cells coming in and eating up the bacteria. Pretty cool. All right, the second type of microscope there on page 33, slide 33, is a dissecting microscope or stereoscope. Stereo meaning it has two, so you can look with both eyes, like a binoculars almost. And then you'd be able to use something like, you'd be able to look at something like this under this kind of microscope, or other types of objects large like this. So 3D structure of specimens has a lower magnification than the other microscope, the first one, the compound one. Light microscope also. This cannot see cells. Easy to use, single lens, not double lens. Light source this time is above the specimen rather than from below. The last one here is from below. That's the light source. Sometimes there'll be a mirror on some of the cheaper ones, but this one is from above. The light comes from up here. And the specimen does not have to be thin. Like I said, it could be three-dimensional and thick like this shell. Dissecting microscope. And so these are the kind of things you can see with the dissecting microscope. So you can see flatworms, large portions of leaves, maybe to see flow of liquids, sap inside of a leaf or something like that. Parts of an insect. This is the ovary of a pepper plant, otherwise known as a pepper. It's like a jalapeno pepper. When you eat a fruit like a pepper or an apple or an eggplant, or a lemon, whatever, you're eating the ovary of the plant. We're going to learn more about that when we learn about plants. And then finally, the last one that we're going to talk about is, I have two more actually, is electron microscope. Okay, these don't use light. These use beams of tiny negatively charged electrons that kill the organism. Organism definitely dead, and they typically coat it with a metal, like even gold. They coat it with a thin layer of metal. Magnifies up to 1 million times, and it's very expensive. So units like this can be $1 million, at least. So you'll only find these in research facilities, in universities, and things like that. But if you go to school for science, or medicine, you can use these for free. You can sign up and use them for whatever you're studying. It's pretty cool, actually. So, electron microscope. And then there's the scanning electron microscope, otherwise known as the transmission. Right? Let me see. Okay, yeah. Can see 3D images. Oh, this is still electron microscope. Sorry. Yeah. Okay. Scanning electron microscope. Looks at the surface of an organism. So here's like a piece of pollen. Here is a flea, a mite. I'm not sure what that is. Maybe another piece of pollen. And again, they cannot be used to look at living things. It creates 3D images, scanning electron. So it's bouncing electrons off the surfaces and making a digital representation, a picture of that. That's what it does. Scans the surface. Whoop. Wrong thing. Come on, Keen, get with the program here. All right, so you can look at things like three-dimensional images of red blood cells. This is a tardigrade, otherwise known as a water bear. And these can live in very harsh environments, virtually no water. They shrink down and become just desiccated, no water, and can live like that for many, many years. And then as soon as they get reintroduced to water again, they start moving around and crawling around. Here's what a cat flea would look like under a scanning electron microscope. Blood-sucking mouth parts. Have been bit by a flea before. That's what the little suckers look like. Pollen using electron scanning microscope. That looks like an orange, but it's a piece of pollen. Pollen is plant sperm. So they release these into the air in the billions, and then they travel around, and when they land on the flower, on the female part of the flower, they travel down. We'll learn more about this, and they fertilize the flower. So. But they come in all different shapes and sizes, and that's where you wonder why people are allergic to these things when they go up your nose, okay? Lining of our lungs, this is like cilia that move that mucus I told you about last time out of that, and it traps debris that you breathe in, for example, pollen. Light bulb filament, which is a metal called tungsten. I actually have some in the periodic table of the elements over there. It's abbreviated by the letter W. Don't ask me why. So anyway, this is the kind of pictures you can get with that. And then finally, sorry, here's the transmission electron telescope. This is slide 43. And this looks at relationships of internal structures. So if you wanted to see the internal structure of, say, a mitochondria, and then compare that to a chloroplast, you can use a transmission electron microscope. And again, can't be used to look at living things. So this is the internal structures of cells and organelles. And then finally, we have staining. So staining, as I mentioned before, when I talked about epithelial cells, like the cells lining your cheek inside your mouth, that's what these are, okay? So, and these are plant cells, obviously, because they're rectangular. So iodine is used in plants, and that's why it makes it this yellowish color. And methylene blue is used in animal cells, and these are cheek cells, and you won't be able to see anything except maybe the lining of it, like the cell membrane. But once you dye it, you can see the nucleus stands out bright and bold. Same with the plant cells. So there's different kinds of dyes or stains used for different types of, because the methylene blue, I don't believe can get through the cell walls, but the iodine does. But iodine is a deadly chemical, so it kills cells. So it's going to kill all these cells. All right, so now you're going to, on the back page, there's a test yourself, so make sure that you do that during this period. And then you're going to label the compound light microscope before we actually do our activity, our virtual activity, because you need to know all these parts, all right? There's actually, I believe there's a question about this on the test, on your next test. So, ocular lens is the eyepiece, and the objective lens is here. Observation tube, the arm, and it's also called the neck. You've got the coarse focus and the fine focus knobs. This is the stage where the slide goes on. This is called the nose piece. I don't know why, it just is. Condenser lens, that's for the light from below, the light source, okay? Base. Iris diaphragm, you turn that to let different amounts of light in, just like your eye opens and closes, the iris opens and closes, your pupil, to let some different amount of light in. So it's all right there for you, and label your notes, and then your notes are done once you do the test yourself. Please go back over your notes and check for blanks, because whenever I go through, I keep looking and I find blanks, and you lose points, and I'm putting in grades like 82 and things like that for notes, and that shouldn't happen, all right? So make sure that you are not turning in notes with blanks on them.